Welcome to today's webinar, which is being delivered by Dr. Kunal Sharan from the Department of Molecular Nutrition of one of our sister institutions, CSIR, Central Food and Technological Research Institute. Kunal did his PhD from CDRI. He did his PhD from my lab. And in 2011, and then he received the prestigious Wellcome Trust Sanger Institute Postdoctoral Scientist Fellowship that year, where he did his fellowship under Dr. Vijay Yadav. And then in 2014, he returned to India and joined CFTRI, where he is currently located. Kunal is, to put it very easily and simply, is that a very prolific young investigator. He received ACRB's Young Scientist Startup Research Grant Award in 2015. In a mapping of Indian research output on osteoporosis published by Annals of Library and Information Studies that recognized researcher with contribution to osteoporosis in India, he ranked second, and he is also recognized among top 5 leading Indian authors with consistently higher citation impact per paper. Kunal serves as an editorial board member of Frontiers in Endocrinology and World Journal of Orthopedics. Kunal has several grants through those extramural funding. Kunal is running his research program at CFTRI. He has a large team of PhD students and has a very busy laboratory. He has 3 patents and he is part of the team that licensed a technology from CDRI. So far, Kunal has published 31 papers with a total citation of more than 1100 and has an H index of 18. With this, I wouldn't take much time and invite Kunal to present his webinar. Kunal.
Thank you very much, Dr. Navidya, for this kind introduction. So, I would be sharing my slides. So, yeah, you can.
Is this your first slide?
No, it's 1 second.
I thought so. It is 27th written. Okay.
Yeah, so you can see the slides, right?
Slide, yeah, good, perfect. Please go ahead.
Okay, so as Dr. Navidya has told that I'm Kunal Sharan, working as a scientist in the Department of Molecular Nutrition at Central Food Technological Research Institute. And today I will be discussing about a topic which is maternal hypercholesterolemia induced programming of offspring's bone development. And it is a new chapter in the developmental origin of metabolic bone disorder. So at CFTRA, what we do is we look into relationship between nutrition and my lab works over bone health. As such, a small group in our department is working on the health outcome which can be affected by or modulated by nutrition. So my area is about bone health. So just to give you a brief background about it. So it is a well-known fact, and everybody must have heard that we are what we eat. Every single cell or every single tissue in our body is made up of the food which we eat. And when everything is made up of the food which we eat and the nutrition which we get from the food, then it plays a big role in our maintenance of our health also. So if there is undernutrition, we are going to have some health problems, whereas overnutrition also is known to have some uh, adverse health effects. And around 2,500 years back, Hippocrates, who is also called as father of medicine, he told that let food be the medicine and medicine be the food. It means at those times also, so much emphasis was given over the food. So this concept is coming from a long time, and recently it is also being said that we are also what our mothers ate, not only during their lifetime but during the pregnancy period also. Whatever mother eats can define our health. One of the earliest examples for that is the Dutch famine, where during 1944 to 45 there was an exceptionally harsh winter and there was bad crops throughout the Europe, and there was 4 years of World War II going on. So during that period, what happened that the Belgium was with the front between Nazis and England. Basically, the two fronts were there who were fighting with each other. The was Belgium chose to go with the England counterpart. So because of that, this German Nazis, they had put an embargo over food transports over their land. During that time, because of harsh winters, there was no crops there itself to eat, and because there was the embargo, the people could not get food itself there. So the people were hungry and starving. But this gave opportunity to some of the researchers later to see how the maternal nutrition during those period, because some of the women at those times were pregnant. So they thought to look into the offspring's health of those females who were pregnant during that time and see how their health was affected because of that critical period during the pregnancy when they were not getting enough food. So the females during that point of time were in their different phases of gestation, early, mid, and late. So first, second, and third trimester, as you can see in the slide. So some women were exposed by famine during late, some were exposed during mid, and some were exposed during early phase of their gestation. So what researchers found later that the offsprings born out of these females, all of them has had glucose intolerance. It means they were all diabetic. They were all found to be diabetic. Whereas during mid gestation, if they were exposed with famine, then they were having microalbuminuria or obstructive airways disease along with this glucose intolerance. Whereas if they were exposed during early phases of their gestation, they were found to have coronary heart disease, arteriogenic lipid profile, disturbed blood coagulation, and increased obesity. So they showed that these critical period of gestation can define the life of the offspring for a long period of time. These symptoms they develop when these offsprings were adults. So the explanation was given later by another scientist whose name is D.J.V. Barker. So he published back-to-back 3 papers in Lancet where he showed that where he gave a hypothesis which is famously known as Barker's hypothesis or fetal programming, where he said that intrauterine signals may optimize the structure and function of organs in the in utero environment in short run, but in long term, uh, if these adaptations are mismatched with the later environment, it may contribute to disease at childhood and adulthood. This is also called as fetal origin of health and disease. So basically what it says that different kind of environment or basically nutrition may affect the programming. Programming is set to be like codes which comes one after another. So in this case, the individual or the offspring is programmed during the intrauterine growth phase itself to have different kind of symptoms at different period or different phases of their life. One of the examples, which is non-mammalian examples in insects, is the African armyworm caterpillar, where or the moth, where the caterpillar, if it develops under nutrient-rich environment into adult moth, what happens that its whole metabolism is uh, carbohydrate centric, and there is no fat deposition in these moths, and they go for short flights. Why it happens is because when the nutrient-rich environment is there, they don't have to go far to get the food. They will get it nearby. So whenever they want, they can eat. Whereas when there is adverse environment or there is a drought, what happens that these moths, they develop into these caterpillar, they develop into the moths which have fat deposition and fat centered metabolism. So fat can be deposited for a long time and its metabolism can occur slowly. So they can go for long flights without eating. So this is one of the adaptations which is seen in the insects itself. Other than that, in laboratory also, Waterland and Juttle had shown that this same phenomena in a genetically modified animal. So if you can see this image, these two mice are genetically identical, but one of them is yellow and fat or obese. The other one is lean and brown. So what is the only difference between the two mouse is that, one's mother, the yellow and fat mother, was fed with normal diet. So they are genetically predisposed to be yellow and obese. Whereas the other one's mother was fed with methylating diet, which contained B12, folic acid, betaine, and choline. So what happened that this gene ABY, which gives this yellow color or brown color to the yellow color to the mice, was methylated. Its promoter was methylated. When what they found was its promoter was methylated, because of which they grew into a brown mice and lean ones. So from this, what we can get out of these examples is that the fetal programming can have several types of effects, like good, bad, and unknown. So the good effects might be used, or we can exploit it to reprogram the offsprings by giving some nutritional interventions to the mother to prevent future disease or have a healthy offspring. Whereas there can be bad effects which, if uncontrolled, some nutritional insults during pregnancy may cause fetal programming leading to future diseases. Whereas some of the unknown effects which we don't know and which we need to study is that during pregnancy there can be nutrient-nutrient interaction, the drug which mother is taking, nutrient-drug interaction or nutrient-environment interaction, which may have some unknown outcome and which needs to be studied uh, to, uh, to, to program our future better in a way, or our next generation better. In our lab, as I told you, we are working on how nutrition affects bone health. So we are looking into different aspects of bone health in conjunction to nutrition. So we look into how maternal nutrition affects bone mass of the offspring during, if we are giving a different modified diet to the mother during their gestational period, how it affects the offspring health. Whereas we also look into how the diet during growing period of the offspring may affect their health, whereas the maintenance of skeleton and during the menopausal condition in animal models, we also look into that, how diet might modify or affect that effect. As all of you must be knowing, but still would like to refresh your memories that bone is bone is a connective tissue which not only gives structure and strength to our body and helps in locomotion, it also harbors certain type of cells and it affects several other functions in the body. So microarchitecturally, bone has two parts. One is trabecular and other is the cortical bone. So trabecular bone is the sponge-like bone, and it is interconnected with each other, and it is mostly found in the long bones at the ends. Whereas cortical bone is present at the diaphysis and it is kind of solid bone. So during osteoporotic condition, what happens that this trabecular structure is disturbed and the bone becomes fragile and prone to fractures. A bone contains basically three types of cells, which are osteoclast, and osteocytes. These are the major type of cells, and they are all interlinked with each other and affects each other functions. So, in normal bone, osteoclast resorbs the old bone, and the osteoblast, they fill these pits by forming the new bone. The osteoclast are derived from hematopoietic stem cells, and their maturation is affected by osteoblast, which secretes factors like rankle and OPG. So rankle, when binds to its receptor rank, it in the pre-osteoclasts, it make them fuse together and become mature osteoclasts, which resolve the bone. And these bone, when they are when it is resolved, through the resolved surfaces, certain growth factors and calciums are released, which act as a chemotactic factor to pre-osteoblasts, which reaches to the resorption site and fills the bone. These osteoblasts, when they lay mineralized matrix, they get entrapped into that themselves and become the mechanosensory osteocyte cells. So this talk is about hypercholesterolemia, how the maternal hypercholesterolemia affects the offspring's bone health. So I would like to give a brief introduction about cholesterol and hypercholesterolemia and effect of cholesterol in our physiology. So cholesterol is a 27 carbon sterol, which is carried by majorly LDL, low density lipoprotein, and high density lipoprotein. Uh, it is produced by liver, and it has several functions. It gives cell membrane fluidity, it makes steroid hormones, sex hormones, and it makes bile acids and helps in formation of vitamin D. During the fetal development also, this cholesterol is produced by the fetus as well as it is transported from the uterus through placenta to the fetus. The function of this cholesterol in the fetus is to make steroid hormones, bile acids, development of brain, whereas it also helps in skeratogenesis through hedgehog signaling and cell growth. So, the maximum daily intake of cholesterol should be within 307 mg. And if you are not taking any cholesterol, that is also fine. The disease which occurs because of increased cholesterol in the circulation is called hypercholesterolemia. And hypercholesterolemia can be of two types. One is familial hypercholesterolemia and the other one is secondary hypercholesterolemia. In familial hypercholesterolemia, what happens that is there is mutation in certain genes, for example, ApoE, PCSK9, LDLR, because of which LDL cholesterol in the circulation increases. Whereas the secondary hypercholesterolemia, the first one, the familial hypercholesterolemia, is a hereditary disease, so it is inherited from generation to generation, whereas secondary hypercholesterolemia is the most common one, and it occurs through diet, drugs, disease conditions, or metabolic disorder. So the secondary hypercholesterolemia is a modifiable disease. The most common reason for the occurrence of secondary hypercholesterolemia is the diet. And the diet which is rich in fat and mostly animal fat is the one which causes this kind of diet-induced hypercholesterolemia. The milk products, meat, these all contains, and the fast food where they use lard, these all contains cholesterol in the diet and may cause hypercholesterolemia. The hypercholesterolemia leads to several other disorders like cardiovascular disorder, atherosclerogenesis, metabolic disorder, kidney dysfunction, neurodegeneration, male infertility, also osteoporosis. So if you see the population wise, the occurrence of hypercholesterolemia is there in all age groups, but its occurrence increases after 45 or 50 years of age. Mostly at later ages, women are affected by it, but nevertheless, there are many individuals, around 8 to 10% of people in the age group of 20 to 34, which usually are at the reproductive age and will be giving birth to the offspring, they are also affected by hypercholesterolemia. So in this study, we wanted to see, because there is a good number of population, like around 8 to 10% females will be affected by hypercholesterolemia during their critical period of reproduction, how this hypercholesterolemia may affect offspring bone health. Although maternal hypercholesterolemia has been shown to affect several other functions like cardiovascular health and atherosclerosis in the offspring, but we wanted to see how whether the bone health is also affected by this or not. So for that, what we did is that we took C57 black 6 mice, and we exposed them with high cholesterol diet containing 0.5% of cholesterol in their diet throughout their gestation and up to the lactation period, when the offspring is getting nutrition through the mother. After that, we switched them into control diet, and we grew them at different ages and saw what happens to their bone health. So the first one, what we did is we checked the body weight of the dams, the pups' weight, their length, the litter size of the mothers, and we found that this hypercholesterolic mother, they could not gain enough weight during their pregnancy or gestation period as compared to the control, whereas the body weight of the newborn pups also was decreased. Even the newborn pups length was less than the control counterpart. What we found that, but surprisingly, is that the number of pups born in the hypercholesterolemic group were more than the control. But however, these pups we were not able to wean these pups because they died during that period due to different reasons. One of the most common reason was caprophagy, because the mother was eating the pups, and we think that it is because the hypertension which develops in the mice when you feed them with hypercholesterolemic diet. So what we did is that we took the day zero pup, the newborn pups, and we checked their bone health by staining them with Alcium blue and alizarin red. So the blue color is given by Alcium blue, which stains cartilage, whereas the red color is given by the alizarin red. So if you can see in these images, the offspring, the high cholesterol offspring, were shorter in size, whereas the craniofacial bones were not properly made. There was a delayed ossification in the intramembranous ossification site in the skull of these offspring as compared to the control ones, and their skull size was also smaller. When we looked into the other appendicular bones, what we found that there was again delayed ossification in the sternum. The forelimb length also was, the forelimb and hind limb length were also decreased, whereas the osteogenesis index, which is the amount of mineralization in throughout the length of the bone, was also decreased. One interesting thing which we found was that there was a fusion of disease in the hypercholesterolemic offsprings at birth as compared to the control counterpart in both forelimb and hind limb. So there was no proper digital patterning in these offsprings. We grew these animals to 12 weeks because we could not rear these animals at different ages. We could only do 12 weeks bone analysis, and we found that in both male and female offspring, when they were even switched to the controlled diet, what happened that their bone mass was reduced. Both cortical and trabecular bone mass was reduced. So the parameters which are directly proportional to the good microarchitecture, they were decreased, whereas the parameters which are inversely proportional, they were increased. Because we could not study the whole pattern of the growth in these animals, C57 black 6, because of bad maternal care and the less survival of the offspring, we switched to another strain of mice, which is Swiss albino mice. Why we switched to this mice is because it is known to have a better maternal care. And similar experiments were done, but at this time we wanted to see what happens at young adult and old stages when the during the modeling phase, during the remodeling phase, and during the bone loss phase in these animals. So we sacrifice them at 6, 12, and 24 weeks of age. What we found is that when we studied the effect of hypercholesterolemia diet on the dams, similar effect was found as we found it in C57 black 6 mice, where the dam body weight could not increase up to the level of control. The newborn pup's weight and length was decreased, whereas again the litter size was increased. But here, in contrast to the previous case, we could wean a good number of pups out of these animals. So similar results were found when in the newborns where the intramembranous ossification was delayed and the appendicular bone or the long bones, their osteogenesis index and length was reduced, whereas there was a fusion of digits in forepaw and hindpaw. When we saw the total growth of the bone at 6 weeks, 12 weeks, and 24 weeks, in both male and female, at both tibia and femur sites, we found a reduction in the bone mass by reduced BMD, BV/TV, and even cortical thickness in these hypercholesterolemic offsprings. So we further did the bone histology in the femur, and we found that there was a reduction in the BV/TV ratio in the in the vertebrae of these offspring. So this we did sectioning of vertebrae and stained them with guancosa. So the black color here denotes the calcified tissue, whereas the blank area is the marrow in between that. So as you can see, the BV/TV ratio was reduced. We also found that there was a reduction in bone formation rate and osteoblast numbers in these bones. Whereas there was an increase in osteoclast parameters like increased osteoclast surface to the bone surface and number of osteoclasts to the trabecular bone area. So we did ex vivo culture to see whether the effect which we are finding in the bone is cell autonomous or it is through some paracrine signaling, because the reduced osteoblast or and increased osteoclast function might be due to the changes in the endocrine factors in the offspring, or there can be cell autonomous programming where the intrinsic activity of these cells are altered. So for that, what we did first ex vivo culture from the offspring of these mice whose mothers were exposed with either control or high cholesterol diet, and we cultured the calvarial osteoblast cells from the offsprings and did several experiments to see their proliferation, differentiation, mineralization, and some critical genes expression. So these osteoblast cells, they first proliferate to increase their pool, they differentiate to become mature osteoblast, and then they mineralize. So for that, what we did first, we did VRDO cell proliferation assay, in which we found that these osteoblasts were proliferating less. Whereas when we did ALP assay, which is alkaline phosphatase assay, one of the markers for osteoblast differentiation, we found there was a decrease in differentiation. By staining also, we confirmed that there was a decreased differentiation in hypercholesterolemic offspring cells, whereas the mineralization of these cells were also decreased. So in conclusion, there was a decreased osteoblast activity, which we confirmed is by cell autonomous effect, because we did ex vivo cultures of these cells, plated equal number of cells, and we found this kind of effect. A gene expression analysis was done to check whether the osteogenic gene expression is also altered because of that, and the cellular effect is as a result of the changes in gene expression, and we found that relative abundance of certain transcript which are essential for osteoblast function, like RUNX2, which is the master regulator gene, which helps in the commitment of osteoblast cells, pre-osteoblast cells towards osteoblast, BMP2, which also helps in the differentiation of osteoblast cells, ATF4, which helps in the maturation of osteoblast cells, COL1A1, which is again collagen type 1, which is secreted as a matrix, osterix, which is another transcription factor. Their all expression level was decreased. We found ALP expression level also decreased. We did Western blotting to find that both ATF4 and X2 expression was decreased and BMP2 expression was also decreased. So these cells were making less proteins which are involved in osteoblast differentiation. We also found that there was a reduction in OPG secretion, which inhibits osteoclast maturation, and there was an increase in Rankel secretion or Rankel expression in these osteoblast cells, which promotes osteoclast differentiation. So this might explain why the osteoclast differentiation was increased. We did again ex vivo bone marrow culture, ex vivo culture from the bone marrow of 12 weeks old mice offspring, where the mother was given hypercholesterolemic or controlled diet during pregnancy and lactation. Later, they were weaned on controlled diet, and at 12 weeks, we harvested the bone marrow from these animals, and we did both osteoblast and osteoclast culture from this bone marrow. So why we did this is because we found that there was a decrease in osteoblast and increase in osteoclast function in 12 weeks or old mice as well. So what we found that by ALP staining, there was a decrease in osteoblast differentiation, there was a decrease in osteoblast mineralization, and when we did ex vivo osteoclast culture, we found that there was an increase in osteoclast differentiation as well. So from this, we concluded that the effect on the bone cells and bone is cell autonomous in these animals, and there might be some paracrine signaling or endocrine signaling also, but majorly the effect is through cell autonomous. So next we wanted to elucidate the mechanism of action, how this kind of effect is happening in the offspring. So for this, we did some literature search, and we found that the cholesterol is very much essential for the activation of hedgehog signaling. And hedgehog signaling is known during the developmental pathway to have an effect in digital formation, the skeleton maturation or skeletogenesis as well. We found that there was a decrease in osteoblast mineralization or activity. The hedgehog signaling is also involved in osteoblast differentiation. We even found that there was a fusion of digits, and hedgehog signaling is known to have an effect on digital patterning. Even hedgehog signaling in the adult animals is known to increase the bone mass or affect the bone mass. The hedgehog signaling, how it works, it either stays in switch off state or switch on state. In switch off state, the ligands, which is sonic hedgehog or Indian hedgehog, they don't bind to the receptor patch, and in those cases, the patch inhibits SMO, another transmembrane protein, and because of that, there is a degradation of gene proteins, and they cannot go to the target, they cannot transactivate the target genes. While when the sonic hedgehog binds to its receptor patch, what happens that the inhibition is lost, and SMO can activate now glyc proteins, and these glyc proteins can go to the nucleus and transactivate the target genes, which affects the osteogenic functions in osteoblasts. So what we did is that we again did the ex vivo culture, and we harvested the calvaria from the offsprings and did osteoblast culture and did Western blotting from them to see what happens to the ligands. And we found that both sonic hedgehog and Indian hedgehog, their levels, protein levels were decreased. And the phosphorylation of the SMO, which is essential for the activation of glyc, was also reduced, whereas Gli expression was also decreased. So from here we found that in these osteoblasts, there was a reduction in hedgehog signaling. What next we did is that we harvested the bones from the adult animals and did Western blotting from the bone of these animals, and we found that there also the sonic hedgehog and Indian hedgehog levels were decreased as compared to the control counterpart. Again, similar to the previous result, we found that SMO activation was not there and G levels were also reduced. So in conclusion, this says that maternal hypercholesterolemia decreases the offspring bone development through inhibition of the upstream and downstream regulatory elements of hedgehog signaling. Next, we wanted to see the experiment which we have done in animals can be can we see the similar effect in the human subjects as well. So for that, we collaborated with Mysore Medical College here in Mysore itself, and we collected the blood samples from both mother and offspring at the time of delivery. So what we did is that maternal blood we collected, and the offspring blood was collected through the cord. So the cord blood contains offspring blood. So through that we collected cord blood and mother's blood. We collected around 250 pairs of MBCP, which is maternal blood and cord blood. And we had some exclusion and inclusion criteria. So the exclusion criteria was that we wanted to see exclusively the effect of secondary hypercholesterolemia on the offspring's bone health. So we removed any cases of familial hypercholesterolemia, CVD or ischemic heart disease or hypertension, diabetes, any other factors which might affect the bone health of the offspring, we removed that. Whereas we took only healthy full-term pregnancies who delivered female offsprings. Because the effect was similar in both male and female, we chose to take only single gender offsprings. So the pairs from mother blood and cord blood from female offspring, which were full healthy pregnancy, were taken. One of the effect of hypercholesterolemia or maternal hypercholesterolemia can be stillbirth or premature delivery. So we wanted to avoid those cases, so we didn't take those cases because we wanted to compare similar age, similar gestational age offsprings, bone formation and resorption markers. So we collected these blood samples and we did correlation studies with maternal cholesterol level and offspring's bone formation and resorption markers. So what we found was that the maternal cholesterol level were compared with or correlated with the osteocalcin levels in the offspring's cord blood. So we found that there was a negative correlation with maternal cholesterol level with bone formation marker, which is osteocalcin in the offspring. Even maternal blood HDL cholesterol and LDL cholesterol gave a negative correlation. Although the correlation was weak with maternal HDL cholesterol, but there was a correlation with osteocalcin. Even PINP gave a correlation, which is another bone formation marker, in the maternal cholesterol with the PINP of the offspring gave a negative correlation, and maternal blood LDL cholesterol also gave a negative correlation. So this suggested that there is when cholesterol is high in the mother or mother is hypercholesterolemic, there is a reduced bone formation in the offsprings. We found no correlation with maternal triglyceride levels with the offspring's bone formation markers. Along with that, we couldn't find any correlation in the bone resorption marker, which is CTX, with the maternal cholesterol or maternal triglyceride, LDL or HDL levels. We then segregated these groups into three, which is normal cholesterol, borderline cholesterol, and hypercholesterolemic mothers. So the diets or the groups of mother blood and cord blood was segregated into this. So normal cholesterol is between 150 to 200 cholesterol level if it is there, it is normal cholesterol, whereas borderline cholesterol is 200 to 240, and 240 and above is hypercholesterolemic. Here also we found that between these group, hypercholesterolemic offsprings, the maternal hypercholesterolemic offsprings had reduced bone formation markers in their blood as compared to the normal and borderline cholesterol, whereas there was no significant difference between the bone resorption marker between these offsprings. So from here we concluded that maternal hypercholesterolemia causes a significant decrease in the activity of bone formation marker in the newborn offspring. So summarizing this result, we found that when maternal hypercholesterolemic diet is given to the mother in mice, the offspring's bone reduces, bone mass or bone health reduces at birth and when they are adult even. And it happens through the downregulation of Hedgehog signaling. We found similar results in humans also at birth. So that was it. I would like to acknowledge my institute where I am. They have given me the space and funding to work, and the funding agencies through which I am getting funds to work on these projects, like from ICMR, SCRP, DBT, and CSR. This work is done by two of my students, which is Ravi Mangu and Kalpana Patel. Without their work, it would have not been possible. So they have struggled a lot. Like Ravi is the one who was the whose PhD work this is. So he had to go to the hospital and collect the blood samples. And because he's a male, he can't go inside the maternity ward. So he had to take Kalpana with him, and she had to go in the labor room itself, and there she had to collect the samples. And they have managed it very well, and they have worked hard on this project. Mostly the animal work was done by Ravi Mangu, and Kalpana helped him in human studies as well as in cell culture studies. So I would like to also thank CDRA because they provided us microcity facility, without which we could not have done the experiments. So that's all from my side. Thank you.
Okay, wonderful, very lucid presentation. I see your institution being painted fresh, or it is an old photograph.
Yeah, it is being painted fresh.
Nice painting, it looks pretty nice, and it reminds me of the old CDRI structure.
Yeah.
Kunal, I congratulate you for this wonderful work. I have to begin with a couple of questions. Number one, do you anticipate intergenerational effect of hypercholesterolemia on bone, the kind of phenotype that you have observed in the F1? Do you think that it will be present in F2 despite the F1 generation's cholesterol level remaining normal? And number two question would be that would you attribute the effect of hypercholesterolemia in bone due to the impaired osteoblast function, because you don't see any change in osteoclast, particularly the CTX level in human cord blood. Don't you think that the effect is basically mediated by LDL cholesterol's oxo-LDL form? That is the one LDL gets oxidized to oxo-LDL, and that can be bad for many cells, including osteoblasts. So is it being attributed by oxo-LDL? So first, if you clarify these two issues, then I'll think some.
So the first question about the intergenerational effect, we don't know, but we think it should be there because The mechanism which we have shown, it's not complete. Right now, whatever we have shown is not complete. Actually, we got a surprising result because cholesterol is known to increase the activity of hedgehog signaling. But we found that it is known to activate hedgehog signaling, cholesterol as such. But what we found was that its signaling was reduced. So what we think is there are two reasons why it can happen, because uh, the fetus also makes cholesterol and it is transported through mother also to the fetus. So what happens that the fetal level of cholesterol is always maintained, whether mother's cholesterol is coming, not coming, or fetus is making cholesterol or not. Most of the time, if reduced cholesterol is there, even then the fetal cholesterol level is maintained. So how it will be happening in hypercholesterolemic case, what we think is that the fetal level of cholesterol is reducing, fetal secretion or fetal synthesis of cholesterol. So that might be affecting hedgehog signal. This is one first hypothesis. The second one, where your question will come, or your question's answer will come, is that there might be some epigenetic changes happening in these hedgehog genes, promoter region of the hedgehog genes, because of which their effect is silenced or the expression is silenced. And this kind of epigenetic changes has been seen to carry for generations. So we need to do this kind of study to breed the offspring again and see whether the second generation, F2 generation or F3 generation also has similar effect. So the strong epigenetic changes has been shown to be carried for 3 generations.
Yeah.
But we could not do it because the mortality of the pups.
Okay.
So we will be planning it later, maybe uh, in future. And to answer your second question, which was that why we could not find any changes in CTX level in the offspring at birth. So what we feel is that during that fetal development or at birth, there is mostly modeling directed bone formation. So the osteoblastogenesis or bone formation is the dominant phenomena which is happening. So that's why we found the changes in bone formation, whereas the osteoclastogenesis is just to shape the bone, not to maintain it. So maybe that's why we could not find any change there. If we follow it up for later, maybe we will find. And for that question, again, in animals, what we found that even at adult stage, that when we did CTX and PINP levels, when we checked, the CTX level were increased. So there was increased osteoclastogenesis in animals at least.
Do you think that elevated cholesterol, because your human data showed that high density HDL was not that much correlated with PINP or osteocalcin, it was low density. So that is why I was wondering that is it all being caused by oxo LDL on osteoblast in case of
That
hypercholesterolemia?
specifically, we have not checked oxo-LDL, so maybe you are right, we can check that.
I mean, I was trying to understand because we have seen data or reports showing oxo LDL causing not only, I mean, you know, the condition of high adipogenesis in the marrow that happens in aging and in postmenopausal condition, and also because of overnutrition, hypercholesterolemia, means the bone adipocytes, bone marrow adipogenesis is increased, and as a result of which they produce LDL and oxo-LDL in the vicinity in the marrow causes osteonecrosis. They kill the osteocytes, and as a result, there is osteonecrosis. And the papers have fairly convincingly shown that it is caused by this oxo-LDL produced locally, but it can be systemic. And in your animals also, most probably the marrow adipogenesis have gone sky high, and it is also contributing to the bone loss.
So we checked marrow adipogenesis. We could not find much difference between, matlab, in sectioning only we checked. We could not find much difference there. The
Oh,
reason being is that this is not high fat diet which we have given. We have
mm,
given high cholesterol diet. Just
high
cholesterol is
cholesterol.
high.
Right, right,
So we have given choline chloride with that, which helps in its uptake and increases the LDL cholesterol in the animals.
right, right. Yeah, yeah. I lost sight of that diet you mentioned. I thought it was just
Yes.
simply okay. Right, good. Okay, wonderful. Any other question? Please go ahead. Doesn't seem like any other question. So in that case, I will thank Kunal again and um, keep joining this webinar. I will announce the next one soon. So please make sure that you and your lab members do participate and join in this webinar. Thank you so much.
Thank you.
Bye.